Thanks very much to Haydeen and Charles and Patty for inviting me and letting me be part of this fabulous symposium and give me an opportunity to talk about my favorite topic, sex in the brain. And for those of you like me with incredibly short attention spans, I've taken the liberty of summarizing the whole talk on the first slide. And it says, admittedly, it's a complex situation fraught with dangerous implications from conflicting interpretations of the facts and the pressure of historical entities. But I'm sure we could come up with something if only I could remember what it was we were talking about. So what I will be talking about is the way in which reproductive steroids can provide spectacular insights into the mechanisms underlying affective regulation and dysregulation. And I'll do that by first sort of introducing you again to reproductive steroids, how they work, with the bottom line being that the effects are entirely context dependent, as is all physiology. And then I'll talk a little bit about depression and specifically the role of reproductive steroids in these reproductive endocrine-related mood disorders. And there again, the bottom line is that there's differential sensitivity to these steroids, and I'll try to talk a bit about why that is. So the main point when you're talking about steroid pharmacology is that structure is function. And you can see that illustrated here. So this is cholesterol. This is the mother of all steroids. These three boxes indicate critical enzymatic modification sites for changing the signaling properties of the compound. And once this 6-carbon side chain is cleaved, you produce pregnenolone, which gives rise to all the biologically active steroids, the androgens, the estrogens, glucocorticoids, mineralocorticoids. And these enzymatic steps are performed by a very small group of enzymes with sort of redundant actions in these steroid cascades. Now, it's really critically important for following the rest of the talk that you remember every one of these structures, because otherwise, keep up. But no, actually, the main thing is again, just to illustrate that steroid signaling is determined entirely as a function of how the steroid is metabolized. So here's progesterone. Just removal of this acetyl group gives you testosterone. If you reduce this double bond with this 4-5 double bond with 5-alpha reductase, you have dihydrotestosterone, which is a much more powerful androgen necessary to sustain the male reproductive tract tissues. This enzyme, 5-alpha reductase plus 3-alpha hydroxysteroid oxidoreductase, produces androsterone, which is a neurosteroid acting very acutely on membrane receptors. If the enzyme aromatase will aromatize this A ring to produce estradiol, which then works through its signaling pathway. And then additionally, if you add an ethinyl group here to make it biologically active and you remove this methyl group, now suddenly you have a compound norethindrone, which is one of the major second-generation progestins used in oral contraceptives. So just these subtle little changes produce major changes in biology. So how does the structure translate into function? Well, everybody knows it sort of interacts with a receptor. And the steroid receptors all share this common topology, if you will, these domains, the N-terminal transactivation domain, the C-terminal where it binds to the DNA, which is highly homologous across various steroid receptors, and then the ligand binding domains. You see various forms of the receptor, progesterone receptor alpha A and B, estrogen receptor alpha and beta, this being actually produced on 2 different chromosomes. But the idea really in showing this slide is that there, in the receptor, in the commonality of the receptor structure, there's also a great opportunity for so-called off-target effects that I'll come back to. Now, within the cell, as was identified by Elwood Jensen in the late 50s, the steroid receptors, this being the estrogen receptor, are intracytoplasmic receptors. They're not receptors that are on the membrane, like for neurotransmitter receptors and peptidergic receptors. And the steroids are lipids. They enter the cell, they bind the intracytoplasmic receptor, and then bind to a response element on the DNA and initiate transcription of genes. It is a little bit more complicated than that, however. So there are a variety of different ligand-dependent pathways in addition to this canonical pathway. One, these estrogen receptors can tether other transcription factors, and by so doing can influence genes that don't have hormone response elements. Additionally, there are a whole variety of membrane receptors. These include an estrogen receptor alpha, estrogen receptor beta, a G protein, an estrogen stimulated G protein coupled receptor called the GPR. Estrogen can bind to caveolin, and by so doing regulate metabotropic glutamate receptors. And finally, it can also bind directly to ion channels, and this was some data from our lab about 15 years ago. But if you take cells that have no estrogen receptor, and then you bind estrogen to a membrane-impermeable protein, BSA, and you incubate it, you see absolutely nothing. But if you transfect in a slack potassium channel, now suddenly you see the estrogen bound to the cell membranes, and the potassium channel is activated. So potassium channel activation, calcium channel activation is directly mediated by steroids binding. But there are two other elements of this. One is that there are also ligand-independent modulation of these receptors. So dopamine and growth factors can directly phosphorylate the receptor proteins, and by so doing, produce changes in the structure of the receptor in such a way that they become active. So for example, dopamine can stimulate lordosis through the progesterone receptor in the absence of, sorry about that. Dopamine can stimulate lordosis in the absence of progesterone. And then finally, this is really sort of critical, this whole notion of the co-regulators, because the effect on transcription of a hormone bound to a receptor is determined by these co-regulatory proteins. And there are about 350 of these co-regulatory proteins. And here's an example. This is the estrogen receptor. And when estrogen binds, it changes, it flexes the receptor in such a way that this part of the receptor, helix 12, flips. And now you expose a coactivator binding groove. And this coactivator binding then recruits in a whole bunch of other transcription and enzymatic factors that are responsible for activating transcription. Conversely, this is an estrogen receptor modulator, it's like estrogen in some tissues, binds in the same pocket, causes the receptor to flip a different way, blocks the coactivator binding groove, and shuts off transcription of the gene by recruiting in co-repressor proteins. So, as I mentioned, there are about 350 of these, right? So this illustrates a couple of points. Number one, it is the shape of the receptor that determines what the transcriptional effect is. So it's less about affinity and much more about the conformational change that gets elicited. The tetrahydrochrysine binds estrogen receptor beta much more avidly than estrogen receptor alpha, but it's a complete antagonist at estrogen receptor beta and a partial agonist at alpha. So it's really the shape of the protein, shape of the agonist that determines the shape of the receptor, which determines which coactivators or co-inhibitors bind it. And these coactivators and co-inhibitors are highly tissue specific, so that the nature of the response, the strength of the response, and the specificity of the response are all determined by these co-regulators. So for example, in the presence of an androgen receptor protein 70, the androgen receptor will be activated by estrogen. Again, getting at this idea that there are these promiscuous interactions between steroids and their receptors. And so what the shape of this sort of on 3 dimensions conforms to here from these pictures from John Katzenellenbogen. So this is estradiol binding estrogen receptor alpha. That's what it looks like. Look at the yellow. And this is what it looks like when it binds raloxifene. So these are major differences in the conformation of the receptor. Okay. So there are lots of sources of variation in steroid signals, right? Hormone, the levels of the hormones, receptor proteins, the scaffolding proteins, the metabolic enzymes, blah, 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 blah. I mentioned the co-regulators. By the way, the same shape, elicitation of shape applies when you're talking about the receptor binding to the DNA, because you can have single nucleotide changes in the DNA that do not affect the coding sequence for the protein, but that nonetheless elicit a different shape from the receptor, and that will influence transcription. And you've heard about epigenesis from our speakers earlier today, particularly Tracy. And just to underscore the point that that epigenesis is also modulated by reproductive steroids. So the take-homes. Structure is function. Not all hormone preparations are the same, therefore. Just calling something an estrogen doesn't mean it's going to have the same effect in different tissues. Effects are ubiquitous, tissue-specific, and context-dependent. So, in fact, all of physiology is context-dependent. You know, there are developmental stage-related effects. You heard about those again earlier today. Charles Phoenix in 1959, as well as Roger Gorski and Arnold showed that exposure of a female rodent the day after birth to an androgen for 1 day will forever program that brain so that when the animal grows up and gets re-exposed experimentally to androgen, the female acts like a male. It will attack the males. It will and try to mount the females. So there are these critical windows that Tracy mentioned, during which exposure to reproductive steroids really affects the organization of the brain. You also see this at the other end of the age spectrum, right? There are lots of changes that occur with aging. There are changes in cognition. Hi, you'll have to forgive me, I'm terrible with names. Autobiographical, unfortunately. But there are also changes in steroid signaling, not surprisingly, that occur with aging. So here you see young rats and middle-aged rats who are protected. This is their hippocampus, obviously. And when they are treated with estrogen and then subject to global ischemia, the estrogen protects the neurons compared with placebo. You see it illustrated here as well. This is with a new-N neuronal marker. Same thing in middle age, but once you get old, bad things happen. This is obviously measures of apoptosis. Estrogen provides no protection at all, and it's probably related to an increased ubiquitination of estrogen receptors after long-term estrogen deprivation or over the course of the aging process. Sex provides a context. It's what this whole symposium in many respects is about, and presumably reflects different exposures to reproductive steroids, although there are some very interesting behavioral loci that have been identified in the Y chromosome, including one for addiction to death and destruction movies, and my personal favorite, inability to express affection over the phone. So as convincing a positional map of this chromosome is, obviously the effect of sex on brain function is just enormous and multifaceted. Both the external environment in terms of what people are exposed to on the basis of sex, internal environment. Danny already mentioned Jill Goldstein's work on immune function in relation to brain activation. But also remember that our peripheral organs are also sexually dimorphic. The liver, adipose, and muscle tissues have transcriptional profiles in which more than 50% of the transcripts are sexually dimorphic. And so these can result in differences in what the brain actually gets exposed to. But as just an overall summary of the types of sex differences, remember, as Peg McCarthy has shown, there are differences in physiology that can converge to produce the same behavior, right? So, for example, parental behavior in the vole is mediated by different physiologies, but very similar behaviors. And then similar processes can diverge to produce different outcomes, seen with the effect of stress on associative learning, in which the effect on females is much different from that on males. There are also many sex differences, and who the hell knows what they mean. And then there are also the sex differences that are mostly useful for generating papers. People, they say, let's run the data and we'll cut it this way, and we find a sex difference in that. So one has to be cautious in one's interpretation. All right, so we've got all these effects, these context-dependent effects on reproductive steroids, But why do we think that they have any role in reproductive function? And one simple answer is that they affect virtually every system that's ever been implicated in the pathophysiology of depression. So estrogen, for example, regulates the synthetic and metabolic enzymes for all of the classical neurotransmitters, the receptor proteins. As I mentioned earlier, it regulates GABA receptor function, glutamate receptor function, metabotropic glutamate receptor function. There's an estrogen response element on the CRH neuron, and there are clear sex differences in the HPA axis regulation, although different in rodents from humans, which is often forgotten. Estrogen is neurotropic, it's neuroplastic, it's neuroprotective against a whole variety of different stimuli, hypoxia, hypoglycemia, amyloid, A-beta, blah, 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 blah. I can't go through all those. But what I want to focus on is this idea of network dysregulation as a way of taking a step back and saying, how does the brain work? What is the brain actually doing? And the brain is responsible for signal processing and information transfer, right? It's got, these 100 billion neurons. And the neurons really fire or they don't fire. That's their job, is to determine whether or not they need to send a signal down the line. And then a group of these neurons can function together to form what are called networks, usually in distributed brain regions, to perform a particular kind of brain function. Networks can dynamically assemble, so the same brain regions can be part of different networks. And then this is what I want to focus on for a minute, which is that the synchronization of neuronal firing conveys information. And this really shouldn't be surprising because we are completely frequency-dependent creatures. What we hear is frequency dependent. What we see is frequency dependent. So, for example, if I were so if you can tell me what song this is if I sing it. Da, 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 da. Can you tell me what that is? No. How about if I go da, 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 da? All right. So, immediately, your brain looked at the pitch, which is frequency, it looked at the internote interval, and it made a decision. You not only can tell me what the song is, you can tell me who wrote it, you can tell me you got deaf, blah, blah, blah. So the fact is, we are entirely frequency dependent, and our brain reflects that in a variety of different ways. Because when neurons fire together at a particular frequency, it conveys information to the rest of the brain and tells it, in essence, what it ought to be doing. So how does that actually occur? So I'm going to try to provide an example of how it is that nerve cells, which are ordinarily randomly firing, can synchronize. So here are metronomes. They're all set to the same frequency, but they're all firing in their own particular random fashion. So now what this guy's going to do is he's going to enable these neurons to very, very subtly affect each other's activity. Just a small influence on the neighboring metronome. And then what you see in a very, very quick time, quick enough, is that they're all synchronized. If he puts them back down on the table again, they'll desynchronize again. This is just what happens with neurons, because when a neuron fires, it exerts a small effect on neighboring neurons, alters the extracellular ionic environment in such a way that now you can get neurons firing in the same, firing together. And what that does is it amplifies the response to incoming neural stimuli, because all of the neurons share the same refractory period and the same period during which they can experience activation and response to incoming stimuli. So by firing together, they are able to dramatically alter signal processing. And it turns out that estradiol and progesterone, actually, but estradiol in particular, are not only able to influence the synchronization process by regulating GABAergic receptor function, which really sort of tunes the brain, this tonic inhibition which influences the general activation of the brain. But estradiol, I'm sorry for the little, whatever that is, the Parkinsonian pointer that I have. But in any case, oh, there it is, okay. Within minutes of being exposed to estradiol, new synapses form. And depending on whether there are other activity-dependent stimuli, those new synapses will either become permanent if there is a second stimulus, or they will disappear if there isn't. So estradiol then not only results in the synchronization of these neurons, but it actually rewires the brain. It's part of LTP, long-term potentiation, because what estradiol is able to do is to alter cofilin in such a way as to influence actin polymerization and actually build out these new synapses. And so this idea of estrogen regulating network activity and regulating synapses allows me to just sort of talk a bit about why we don't know anything about depression. And part of the reason is we have thought about, some brain lesion in depression. But depression is not a hardware problem, it's a software problem, right? I mean, people get depressed and then they get better. The problem with depression is it's an adaptive failure. It is a lack of integration and orchestration of neural networks in such a way that you get this cacophony that results in a persistent dysphoric state that then can be switched out later on, right? So this then leads to underscores the notion that we should be thinking about depression in terms of its network activity. And there is lots of data accumulating suggesting that a variety of the different networks, a default mode network, interior thinking, sort of reviewing your past, daydreaming, rumination, social cognition, reward networks, that there are dysfunctions in all of these networks that have been observed in depression. And then that brings the second beef that I have with the way we think about depression, which is it's a bunch of symptoms, right? This is this whole ridiculous DSM mindset that, 5 of 1, 3 of these, congratulations, you're depressed. When in fact, what we need to recognize is that these are state-related disorders. Now, what do I mean by a state? A state is a self-organized ensemble of different kinds of ideas, affects, memories, self and object relations. It's sort of a program for interacting with the world, for interpreting events. We all have these states, right? I mean, you could be saying, I got to see this patient, and, she's just going to tell me I don't know what the hell I'm doing. I'm in Boston, and, the traffic is so horrible, the cab drivers have to catheterize each other in the morning, and the weather is, how can anybody live here? And I don't like whom I'm living with. And then something can happen. You have a great experience, go out to eat with somebody, a sporting event, sex, drugs, rock and roll, back in the same spot. And you go, hey, I have done better with this particular patient than all of her prior therapists. I have this unbelievable ability to help people really integrate their experiences in such a way that they can increase their personal freedom. In Boston, a fabulous place, the most intellectually vibrant place on the planet. So you call that a change in perspective? No, it's a state change. The way you will respond to the world and to yourself are different. So the advantage of thinking about these state changes, first of all, is it puts these disorders on a spectrum with normal behavior. We all change state, right? The problem with depression is that they can't. They get into a dysphoric state and they can't get out of it. That's part of the despair of depression. No matter what they do, they can't get out of it. Panic anxiety syndrome, dissociative disorders, these are all OCD, all state-related disorders. But it also allows us to focus on the kinetics of how we change state. This was talked about like 50 years ago, that you need to focus on when people switch, not the contents of the state itself. So if you look at women with PMDD, whatever it's called, I can't even know what it is anymore, premenstrual dysphoric disorder, these are mood ratings, and what you see is, they get more and more symptomatic, and then there's this sudden switch out in these 3 patients. So maybe we can recast the symptoms of this disorder instead of thinking about, they're staying at home and disturbed, social, reinterpreting things in ways that seem to be particularly dysphoric. Social cognition abnormalities, salience network, affective regulation, a reward network. So we could ask then whether these networks are regulated by reproductive steroids. And here are just several examples. This is data from Peter Schmidt. Peter and I have used and this, by the way, is also from Karen Berman, used a model in which we administer leuprolide acetate, we shut off ovarian function, then we add back estrogen in isolation for 4 weeks, and then progesterone in isolation for 4 weeks. And here you see 2 nodes of the default mode network, the medial prefrontal cortex and the precuneus. You see them substantially more activated under estrogen than leuprine-induced hypogonadism or progesterone. This actually is data going way back to when Danny Weinberger was a very young human being at NIH, working again with his fellow at the time, Karen Berman, and showing correlated activity between the prefrontal cortex and the hippocampus under conditions of estrogen replacement or progesterone replacement. You see this connection, but they become disconnected. The activity loses its correlation under conditions of hypogonadism. And then in terms of the reward circuitry, my former fellow, Jenna McHenry, is now professor at Duke, working with Garrett Stuber, focused on neurotensin neurons in the medial preoptic area. And what she did is she did a number of studies, one of which was an optogenetic study using virally mediated transfer of channel rhodopsin into these medial preoptic neurotensin neurons, hooked the fiber optic cable up to a lever so that the mouse could self-administer the light hooked up to these neurotensin neurons. And it delivers a 3-second light. And so this is a mouse during proestrus. And you just see that she can't get enough of this. Illustrated here during proestrus when estrogen levels are high, she'll do this for 1,500 times over the course of 30 minutes. So estrogen is profoundly rewarding under these circumstances, and whoa, that's cute. And I don't know where that even came from. It's not even on the slide. So let's just for a second. But what she also showed using in vivo 2-photon calcium imaging is that these same neurons were not only highly rewarding, but they were specifically rewarding to sexual stimuli. That is, the odor of a male would do it, appetitive stimuli, food wouldn't do it, and was also associated with striatal dopamine release. So this is a medial preoptic area, GABAergic neurons going to the ventral tegmental area, and then promoting dopaminergic activity in the nucleus accumbens. So the reward circuitry, there we go. So we got, okay. So these reproductive steroids then are capable of influencing the circuits that have been implicated in depression. But do they play a role in reproductive endocrine-related mood disorders? And if so, why do only some women get these disorders? Because this is really the critical question, cutting across all of the disorders we study. Not everybody gets, no matter what it is that we're studying. So what's the source of that vulnerability then? So going back again, I lost several slides here. Oops, that's not going to work. Sorry. Okay. So first, about 20 years ago, Peter Schmidt gave estradiol to women who had perimenopausal depression, showed that 3 weeks of estradiol was a very effective antidepressant, whereas placebo wasn't. But that doesn't tell you that estradiol is at all related in the ontogeny of of perimenopausal depression. So what we did is looked at women who had a past history of perimenopausal depression, blindly put them on estradiol, kept half the group on estradiol, and blindly withdrew the other group. And what Peter found with these weekly mean mood ratings is the group, not surprisingly, kept on estradiol, did fine, and those who were withdrawn got depressed almost immediately before the onset of hot flushes. So this makes it look like estrogen withdrawal is a trigger, but this same exact design used in women who had no history of perimenopausal depression had absolutely no effect on their mood. So it suggests that this hormonal stimulus is sort of a source of differential sensitivity in a certain group of women. We found the exact same thing in women who have a history of postpartum depression, and we found the same thing in women who have PMDD. So these are weekly mean mood ratings. You see the symptoms in the premenstrual week. If you suppress ovarian function for, by the second and third month, the symptoms are gone, as contrasted with placebo. Now you take this group and you add back estrogen and progesterone, and now you see, again, very rapidly, you see a return of depressive symptoms. So this looks like these hormones are causing the problem, but if you do the exact same study in women who have no history of PMDD, absolutely no perturbation of mood at all, right? So the hormones are causing the symptom. There's nothing wrong with hormone levels, incidentally. But the hormones are causing the problem, but only in a select group of women. Now, this could be attributable to the fact that you're now causing an increase in hormones, right? So it's a change in the hormones that's precipitating the signal. Or it could be that you need to have a certain requisite level of hormone interacting with an infradian monthly Zeitgeber that results in people getting depressed on a more or less monthly basis. So what we did then is to develop a study in which we suppressed ovarian function for 5 months, and then for 3 months just added back continuous estrogen and progesterone. If it was due to a monthly sort of Zeitgeber, you would see that patients would get depressed and then they'd get better, and then a month later they'd get depressed and better. If it was due to the change in the hormone, then you'd see that they'd get depressed and then not thereafter. How many people think it's due to the level in the hormones? Nobody. One person? Nobody. How many people think it's due to the change in the hormones? 8. How many people have no active cortical function at this point? The 8 are absolutely correct. So here are the symptoms at baseline. This is the absence of symptoms during ovarian suppression, absence of symptoms during placebo replacement, and then you add back the hormones, and the patients get depressed, and then no more depression over the remaining 3 months. Now, obviously, there's some variability in the latency following the stimulus, but if you want to look in one person, these are what the typical symptoms look like. So the change is the signal, right? But only in the context of susceptibility. The question that we have then is, do these reproductive steroids differentially affect brain regional activation, right? We can see that there are these behavioral effects. Is there any evidence that there's something in the brain response that would correspond to that? And I wouldn't ask the question if the answer were no. So here again, using the Lupron model, looking at these 2 brain regions that are critically involved in affect regulation, controls are absolutely basically showing no difference at all in the hormonal state, whereas the patients with a history of PMDD show dramatic differences, real reduction on progesterone in both these regions relative to estrogen replacement. So remember, however, that progesterone and estrogen aren't the only things that change over the reproductive cycle, right? There are also these metabolites of these hormones like allopregnanolone, which is a ring A reduced metabolite of progesterone, and which is a positive allosteric modulator of the GABA receptor. So what has been, I don't want that one. Oh, God. No, that's all right. The allopregnanolone is unique in some respects as a neurosteroid, acutely modulating GABA receptor function, because it dials up these particular subunits, these delta receptor subunits. So the delta receptor subunits are extrasynaptic, are part of extrasynaptic GABA receptors that, again, are responsible for tonic inhibition, as opposed to the synaptic GABA receptors that are responsible for the phasic inhibition when you get an action potential. So during pregnancy, during changes in hormone, you get actually a reconfiguration of the GABA receptor so that you get more of these alpha-4 and delta subunits, increasing the sensitivity to these neurosteroids. And by the way, benzodiazepines don't bind to these receptors. So we asked the question, could changes in allopregnanolone influence affective state? Is there something about these changes in allopregnanolone which you would get with changes in progesterone that occur over the course of the menstrual cycle, because it's just a metabolite of progesterone? So could these changes be responsible for some of the symptoms that we see? And so Pedro Martinez, working with Peter and me, developed this study using an alpha-5 reductase inhibitor called dutasteride that prevents the formation of allopregnanolone. And what you see is that these are the baseline profile of symptoms. This is what it looks like on placebo. And you see that the symptoms were quite dramatically reduced in those women who never showed the increase in allopregnanolone that ordinarily occurs following ovulation. And then as well, these neurosteroids have recently been used for the treatment of postpartum depression. Here's a study in which women performed by Samantha Meltzer-Brody, showing that women with hospitalized for postpartum depression get an infusion of this neurosteroid. Within 24 hours, their depression is gone, and it doesn't return to baseline, despite the fact that the infusion is only lasting for 5 minutes. So, no, sorry. The infusion is 60 hours, but I had 5 minutes. I was just, working with it. But you can also see that the allo successfully beats placebo in terms of response rates and remission rates, which are really quite high. All right, so we got to the last two questions. What causes the susceptibility, right? And when you think about the susceptibility, obviously we heard this morning, it's all about genetics. And when I think about psychiatric genetics, I think about this, Jimmy, sixth generation pain in the ass, these heritable behavioral traits that get handed down over the generation. But what I'd like to focus on really is this possibility that we can find genetic correlates of this hormone sensitivity. And so Peter, working with Nalima Dubey and David Goldman at the NIH, took women who were part of our Lupron study and who showed hormone sensitivity in that Lupron study, developed lymphoblastoid cell cultures, and then either just compared them to controls untreated or following exposure to estradiol or progesterone. And then, once the pathway analysis was performed, this study was replicated in another group of women with PMDD and control using qRT-PCR. And as one example of the pathways identified, which to me is very, very interesting, is this ESCZ pathway, which is part of the polycomb repressor complex that is responsible for these epigenetic methylations that you heard about. And these tend to almost all be upregulated, about half of them significantly so, in both the original and the replication study in the women with PMDD. And then further, the changes in progesterone that the cells see. In other words, when they're exposed to progesterone for 24 hours, the cells, the control cells, show an upregulation with progesterone that is absent in the cells from the people with PMDD. And just two more examples of that, but this with a group with perimenopausal depressed group. That is, they're not depressed now, they have a history of perimenopausal depression. But Sarah Rudzinskis, working with Peter and David, doing a similar cell culture using iPSC, NSCs actually, and lymphoblastoid cell culture, showed an upregulation of this particular metabolizing enzyme, 7-alpha hydroxylase. And you see it under all three conditions compared with controls. But what was particularly interesting is this chemokine CXCL10, which is critically involved in heart failure, which is greatly increased in women following the perimenopause. And here you see not only an upregulation in the cells of women with past history at baseline and following estradiol, but particularly upregulated following estradiol withdrawal. So that brings me to the last point, which has already been mentioned, and I think very elegantly, that it's really about this interaction between environment and genome. And I just want to highlight two studies by Jamie McGuire, who with Istvan Modi, about now 10 years ago, did this wonderful study in which she looked at delta subunit knockouts in the GABA receptor. And what she found is that these animals, and this is critical, at baseline were perfectly normal. They grow up, they look fine, but once they deliver, they look depressed, they're anhedonic in the sucrose preference test, increased immobility for swim test, greatly increased stress reactivity, and most notably, they wind up killing their offspring. So here you have a genetic abnormality that only expresses itself in the context of a reproductive endocrine change. And she subsequently has an even more elegant animal model of this. This is the last slide. Subsequently, more elegant animal model of this, looking at the potassium chloride cotransporter and showing that in the absence of that in CRF neurons, she has the exact same phenomenon. So, okay. There. So, this is obviously a fairly complicated matrix we're trying to understand. But if we approach it not as illustrated in this page from the Annals of Equine Medicine, where irrespective of the disorder, you just treat it all the same way. I mean, that's what we're learning. There isn't going to be a pathophysiology. There isn't going to be a gene, as Danny said. But if we just keep that in mind, you know, we'll be in the right place at the right time to really be able to take these wonderful insights that we're deriving from neuroscience and use it in a way that we can translate into improved health for our patients. Thank you very much.
Great, thanks. Maybe just a couple of quick questions, and if you could also try to keep the answers short so we can stay.
Is that even possible? Yeah. I can do it afterwards.
Yeah, plenty of time afterwards.
Yeah, sure. Just hook.
Go ahead.
Okay, thanks. Have you thought about looking at any of this in people who are receiving hormone treatment for
I'm sorry,
sex
people who?
change? Receiving hormone treatment for sex change, transgender.
I haven't. There are people, Neil, I imagine that Neil Epperson is at the University of Colorado, but no. Yeah, there are, I'm doing what I was told not to do. There's a Dutch group that did this many years ago, but they published a number of papers on it, but not using modern techniques.
One more.
Thank you, David. Question that I'd asked you before about differentiating steroid hormones from the periphery versus the neurosteroid, trying to disentangle what may be centrally based versus peripherally driven.
Yeah, so that's a wonderful question, and it's not a short answer. I mean, and this is what we talked about. I mean, I think, you know, it is perilous to infer that what's happening in the periphery is somehow driving what is taking place centrally. It is likely a combination of the two, but I do think that using peripheral measures in combination with system level outcome measures will enable us to decide whether these peripheral measures really do account for a substantial part of the variance. Because otherwise we're really sort of left with, you know, gross changes in brain imaging under cognitive activated conditions or with behavioral changes, which is really not satisfactory.